This talk will be primarily going over the paper discussed, integrated informatics analysis of cancer-related variants with OpenProfile. So the talk can be broadly grouped into two sections, but there's one overarching theme, which is that variant classification tasks, in particular for this talk in the context of somatic variation in cancer, can be streamlined with the use of variant annotation software. This can incorporate things like predictive methods, but can also incorporate other things, including genome annotations and known clinical relevance. So the first basic part of the talk is introducing the OpenCravat software, what it does, why it's important, and how it can help various variant analysis tasks. And the second part is a series of case studies to illustrate how annotations and in particular OpenCravat can be used to find novel findings from variation data without overburdening the user with too much coding. So we'll start with an introduction to OpenCravat. What is it? Well, it's a Python package, basically, that performs variant annotation. There are several tools out there like VET and Annobar that do variant annotation, but there are some drawbacks to the way that they do it. For starters, OpenCravat is open source and locally installable in an easy way so that folks in industry and academia can download this tool, use it to their heart's content without worrying about how to put it on their system or any of those kind of cumbersome software associated tasks. And what makes OpenCravat unique is that it's a platform for not only doing variant annotation, like take in and input variant file and return annotated variants, but it's a platform for sharing and integrating annotations that can be used in analysis. There are 4 basic steps to this process. And what makes OpenCravat stand out is that it has an app store of annotations. So the first step is to select the annotations that are relevant to your analysis from the OpenCravat app store. We are primarily funded right now through a cancer-related grant. So we have an app store of different annotation resources. Many of them are relevant to cancer. Known driver genes, predictions of if variants are likely to be drivers, and things along that nature. But there are other kinds of annotations, and I'll get to them in a moment. So once picking out the annotations that are relevant to the analysis at hand, the next step is to annotate the variants. So that would entail a file that contains a list of variants and giving that file to OpenCravat so it can return not only those variants, but the genes they're in, the elements they impact, and what annotations are relevant to those variants. The third step is sort of optional, but OpenCravat features a graphical interface that can be used to pull out the variants from the data and filter them based upon their characteristics to really quickly find those that are most relevant to your analysis and set aside the rest until you want to look at them. The fourth step is, of course, to export and share the results. So multiple different kinds of output files can be generated, and even the entire interface can be ported and sent over to a different user so they can see exactly what you're seeing. So first off, what is the App Store? What is it really? For folks who aren't super computationally savvy, we spent a lot of development time creating a front end that is biologist and user friendly. So much like many other app stores that have been seen, it's got a graphical interface with a tile representing each app that is available. And you can see the categories along the left represent the kinds of annotations that could be used, like 1,000 genomes allele frequency or nomad allele frequency. There's the CAD predictor. There's also the VEST predictor included in here. But beyond annotation sources, there's also more complex things, like we see the 23andMe converter that allows users to input 23andMe formatted files. There's also base changes, which is a graphical plugin that lets users see what kinds of base changes are in their variants of interest. By selecting only these things that are relevant to the work at hand, it can really simplify the analysis task downstream. So set aside predictors that aren't interesting, allele frequencies that aren't helpful, like populations that aren't relevant to the sample at hand. The formation of this app store means that the software is somewhat variable. Once you've installed the base package, you then select the apps that are actually relevant to the work that you do. And then it's not many gigabytes worth of information. It's only the curated subset of what's relevant. And doing so allows us to make an app store that includes, I don't know, more than 150 things right now. And that really does help with very specific analyses. To actually run OpenProvat once all of the annotations are kind of sifted through, it's relatively straightforward. I've included an example here on both the command line and in the graphical interface. And these are both packaged together. So you can use one or the other or switch between them like I do. So the command for running an annotation job is fairly straightforward. On the command line, it's OC run, then you tell it the input file, and you tell it the genome version, and you select the annotations that are interesting. Here I use the ClinVar database. And then the -T says, what kind of output would you like? So this command is adding ClinVar annotations to the variants in this file and outputting an annotated VCF. Pretty simple. It's similar in the graphical interface. You select the genome version, you choose your file, you click the checkboxes for the annotations that are relevant, and then click the big annotate button, and your task is basically done by that point. The table on the right shows previously run jobs, so you can see how big they were, how thickly you annotated them, and even download extra files. Well, that was step 2, annotation. It's really straightforward, right? Well, the next step is the interactive step of digging into the data and finding the variants that are most interesting and relevant. That can be done outside of the interface using command line tools if desired, but we've had a lot of fun creating an interactive results viewer for this kind of work. So in the GUI, you click Open Result Viewer and it pulls up the result viewer. Here's an example of what it might look like for an example job for the variant tab of the interface. It's divided into 2 components here. The top is a table where each row in the table is a particular variant in the input file, and every column refers to some annotation source. Here we see ClinVar in the middle with clinical significance, reference numbers, disease names, really dense information. But if ClinVar is really being used, it's all pretty necessary. For example, conflicting interpretation variant with only 2 submissions is a little bit less interesting, let's say, than a pathogenic variant with 5 separate reports without any conflicts. So by using this interface, it's relatively straightforward to pick a variant of interest. The bottom half of the page basically takes the information shown in the table for that row and expands it for a more easy to view interface. So here we have selected the CYP1B1 variant, and in the bottom half, we can see the chromosome position, ref alt. You can see that it doesn't have an annotation in Cosmic, and we can see that it has several interactors in BioGrid. It's easy to copy and paste that. You can see, for example, on the right, there's this Nomad widget that indicates it has no presence in any of those allele frequency populations. And even we can see a glimpse of a network diagram in the bottom left. But it's oftentimes not just looking through every single variant exhaustively where the work happens. Filtering is really the bread and butter of variant annotation, and it's these annotations that are used to filter variants. For example, this filter tab of the graphical interface here I have shows a population allele frequency of less than or equal to 0.1. Now, your exact threshold may vary, but for the purposes of illustration, we can see how for, say, a random human genome of germline variants, it would be very helpful to, before digging into any analysis, first filter out, let's say, variants that are too common, variants that aren't coding, variants that aren't predicted to be pathogenic. These 2 components together make a really powerful tool. But beyond being able to explore the data visually rather than on a command line, there's also the visual component of graphics that we can now add. And I'll run through some of these examples. The real benefit of this is that it shows many variants at the same time. For example, this at the top left is a sequence ontology summary. It's on the summary tab of the interface. It provides a quick glance at what kind of variants are in the input file that has been taken into consideration. If it's many somatic variants, it would then be really easy to tell which are missense, which are indels, and get an overall sense of the data. The bottom left diagram is a protein diagram for a gene in particular. And what it does is shows a visual representation of a protein sequence, and the yellow thermometers indicate where variants lie along the protein sequence. If we had many folks with the same type of cancers, it would be apparent that there might be clustering at particular hotspots. And then that information could be taken to better study that particular clustering of variants rather than all variants simultaneously. But it gets even more advanced than what are the variants and where are they? The top right shows the rank score widget. This shows the 4 variants in the particular gene I have selected and contrasts the pathogenicity and evolutionary prediction scores for the different variants. That way we can not only see whether or not a given variant is predicted to be pathogenic, we can see whether there's agreement between different methods and if there is more systematic bias happening here. So ClinPred thinks that none of these 4 variants are particularly pathogenic, but Revel on the far right thinks that maybe the one at the bottom right could be pathogenic, but the others are probably not. In this way, there's really complicated information, pathogenicity prediction scores that myself and Dr. Karchin have created that we want to put out there, but they're pretty hard to interpret in isolation. So by building this kind of interface and this annotation tool that brings it all together, it makes our tools even more useful than they are in isolation. An extra benefit of having a tool that has a plugin system is that you can have plugins of analysis tasks. This is an example of the case control analysis plugin. In this setup, a user has, let's say, a multi-sample VCF file that has some samples with a phenotype and some samples without the phenotype. By adding an extra file as an input to OpenCravat, here defined as just the patient IDs and then the word case or the word control and a simple tab delimited file, it's possible for OpenCravat to do a little extra work on the back end and count how many cases and controls defined by that file are heterozygous or homozygous for each variant in the file, and then compute a very simple Fisher's exact p-value based upon 3 different kinds of inheritance models. By doing all this kind of basic data processing on the back end, it makes it really easy for users to quickly sort and filter and pull out the relevant variants fast, and then get into analyzing those rather than the entire set of data. And okay, one last plug about OpenCravat is that it runs on several platforms. I have it installed on my MacBook Pro. I have it running here most of the time, but I also have a Windows computer that I have it installed on. For my MacBook, it's really easy to install with Python pip. You just do pip install OpenCravat. But for my Windows machine, I use the installer. You just click next, next, next. Really easy to use. It's also possible to run via an internet browser. If you go to the World Wide Web and type in run.opencravat.org, you can put in files up to about 1 gigabyte in size. And so long as the data isn't private, the annotation can occur there as well. It's also running on departmental multi-user servers and on NIH's FileWolf, in case anybody has access to that. And for extremely large jobs, we have worked, teamed up with Microsoft Genomics to port OpenCravat to Azure. Please reach out to me if you're interested in working with them. And we also have it working on Google Cloud and on AWS. So OpenCravat can go wherever your data is and scale to the needs of the data. If it needs to be maintained privately, you can put OpenCravat on the server where the data is. And the only time OpenCravat will touch the internet is to download the databases from the App Store that you've decided are relevant. Your data never goes up and touches the internet. That's not safe. So now that I've plugged OpenProvat quite a bit, let's get into how variant annotation in general and OpenProvat more specifically can be used to help with variant prioritization in the context of cancer. And for the first case study, we're looking at variant prioritization in multiple lesion cancer samples. So as I've established, the point of variant prioritization is to find those that are most worthy of looking into. So one strategy of doing so in the context of cancer is to find clonal driver mutations. Those are the mutations that occur in the initiating tumor cell and are inherited to most of the other tumor cells if we just kind of do a scan. So this can be a task. Given multiple samples, identify variants that are in all of the samples and see if they are driver mutations. So we have this information from a study indicated at the lower right. They have high-grade serous ovarian cancer. And we were lucky to have samples from these folks available to use through EBI. They were sequenced in some depth. They have somatic samples from tumors in the left and right ovary, 3 metastases, as well as the stick and the p53 signature. But goody, we also have a normal DNA sample. So now the task is relatively straightforward. We find the mutations that are in all of these, or most of these affected samples, and not in the normal sample. But let's go through the steps of OpenProvat. The first step is pick the apps that are relevant. And we're stepping through it like I've never installed OpenProvat before, and I definitely don't have any of these pre-installed. Right. So the first thing we could include is something called cancer gene census. This is how the store interface looks as shown on the right. So I'm going to stop sharing my screen for a second. We're going off script. When running OpenProvat, the App Store is available right here. And by clicking on the tile, it pulls up and a really quick but clever introduction to the tool in case you've never used it before. Like this screen database is searching for cis regulatory elements, and it's from the ENCODE database as information like the website to go get the data, the paper in case you use the data in your analysis. This is what you cite. It's all ready to go, as well as the size of the data and a basic introduction to it. So for this case study, we typed in cancer, and one of the first things to come up was cancer gene census. It's a publication from 2004, and it's basically a census of cancer-related genes. Well, definitely, if we're looking for driver mutations, we should look within genes that are known to harbor drivers. We could also include another list of cancer driver genes or oncogenes and tumor suppressor genes established by Vogelstein et al. This was back in 2013. So by installing these annotators, OpenProvat would then say, oh, this variant is within an oncogene as defined by Vogelstein et al. Okay, what else can we include? Well, another good resource is something called CASM+. It's a predictor also made by the Karchin lab that takes a missense variant and determines if it is more likely to be a driver mutation or a passenger mutation in the context of human cancer. It returns a p-value that can be used to kind of gauge the certainty of this prediction. And it has this added benefit of having multiple sub methods within it that have been trained on particular cancer types. So I've written CASM plus OV because it's the CASM plus ovarian predictor that we can be using for this example. Very exciting. But we'll also include a couple of general use tools. The first is MutePred. It's a very basic pathogenicity predictor outside of the context of cancer, but it has this extra feature where it not only predicts pathogenicity, it predicts the impacted molecular mechanisms by the variant. So this might give us an insight into what's happening, what is the variant doing in its protein. There are other tools that are available to be used for variant effect prediction. We've got, I don't know, maybe a dozen or so right now. Of course, Best4 is something also produced by the Karchin lab. It works pretty well, and I would advocate for using it. And we'll also include in this analysis the IGV visualization tool, mostly to plug that it's available as a plugin for analyzing bands. Okay, so now we have inputted to OpenProvat all of those variants from the different samples, as well as those annotations of interest. We're going to use the filter interface to narrow in on variants that we know are interesting. In the filter interface, the first section of it is called samples. So if you have in multiple files, you can select variants that occur in some files and not the others. The red X here indicates that we are excluding variants that occur in the normal VCF. Very easy. The bottom section of the filter tab allows for the creation of incredibly intricate queries. So this first segment of the query defines that we're looking for variants that are missense, splice site, frame shifting, and non-frame shifting endouts. The second part of the query is a little bit more intricate, but it's basically an OR. It's saying, is this variant within a gene that is either an oncogene or a tumor suppressor gene in cancer gene landscapes or cancer gene census? And to apply these filters, just click the Apply Filter button, and very quickly, we've gone from 139,000 potential candidates to 9 strong candidates. So what are they? Of them, we can look at how many samples they're in and see which of them are likely clonal. And 2 of them are observed in 7 of the 8 lesions, so they're probably clonal, one in RANDP2 and one in TP53. If we go back to the original study, we can see that they did deep targeted sequencing and did verify that the TP53 mutation was in the 8th lesion. Sometimes variant calling isn't perfect, so we have to have a little wiggle room here. The TP53 mutation is a known driver. It's predicted to be a driver by CASM+, and it's predicted by MutPred to be a mutation that results in loss of sheet structure. So in the case of this patient, we would say focus in on the TP53 mutation as a likely driver and see what kind of treatment options spawn from there. We were also able to use the IGV widget to verify that the TP53 mutation was present in the BAMs it said it was. That's all good. And the index network widget, which I'm just mentioning because I think it's really cool, found 13 networks in the NCI pathway interaction database relevant to TP53, as might be expected. TP53 is pretty pivotal. All right, let's move swiftly on to case study 2. This is a completely different kind of exercise. The first exercise was 1 person, multiple samples. Case study 2 is multiple samples, multiple people. In this study, we'll be identifying driver missense mutations among many, many metastases. So this terrific paper by Ryder et al. Sequenced untreated metastases from 20 different patients with a variety of cancers. And they were able to pull out 15,765 missense variants among those folks. So we have a TSV file they have provided with many, many missense variants. And our goal here is to identify the driver mutations that we can identify at least from that TSV or prioritize them. For a quick example, I'm going to show how this works on the command line interface to do the module installation and running OpenProvat. So on the command line, if you'd like to install regular CASM+, because this is many kinds of cancer, you do OC module install CASM+. We're also installing the TSV reporter because we're going to want to output a TSV file. So this should look familiar. Running OpenProvat is OC run. The input file, we're naming the file writer 2018, outputting a TSV, and the file is in HG19 format. If I don't say myself, that is a pretty straightforward query for a command line. We're trying to make it more accessible for biologists to use the command line. We did an extra step at the end. Since CASM+ outputs a p-value, we did a multiple test correction to provide a q-value for each mutation. So that's a little bit of work behind the scenes. So what are these variants that among the 15,000 that look suspicious like they might be driver variants? 56 of the 15,000 were predicted as drivers by CASM+ with a significant adjusted p-value. And many of them include things that we already know are drivers, like KRAS, SMAD4, and PTEN, R173H, kind of hallmark things. So that justifies that CASM Plus is doing a good job at finding driver variants. And here we have just an example of how the protein diagram might be used in your work if you've got multiple samples. This RAS domain at the very front looks like there's a couple mutations occurring at the same place. It's a little bit easier in a graphical display to quickly see a pattern within a gene rather than filtering through all the variants and seeing the numbers. The index widget was particularly helpful in the case of the Writer et al. Data because it included multiple folks. So the red dots indicate that this is a network that has 2 genes mutated in this data, and it might indicate a pathway that is recurrently impacted by drivers in different folks. So that was case study 2. Multiple samples from multiple people, putting it all together and drawing out new inferences. So let's turn that on its head, and let's just take one sample from one person, and let's make it germline. In this example, we'll be using annotations to find clinically actionable germline variants from a single human genome. It's more of a thought experiment, really. Given a single human genome, can we really pull anything out that's useful, particularly from the cancer perspective? We identified a person in the Personal Genome Project and got their permission to do this study and annotated their variants to find things relevant to cancer. We used 3 different kinds of annotations here: the ClinVar database of variant disease associations, the PharmGKB database that includes variants and their pharmacological relevance, and then something else called ClinGenGene, which uses the ClinGen curated gene disease associations to highlight variants that might not be known already to cause disease, but occur within genes associated with diseases. So what do they return? Well, ClinVar found dozens of variants with some annotation. And among them, more than a dozen is actually relevant to cancer, but let's go through what they are. There is 1 protective variant for lung cancer, 2 variants known to be risk factors, 5 variants that are called pathogenic, but are non-coding related to AML. There's another pathogenic intronic variant associated with a hereditary prostate cancer. And then finally, at the end, there's 16 different variants that are known to be related to drug response with respect to dosage, efficacy, drug reactions, and things like that to various cancer treatments. So lots of information pulled back by ClinVar. Next, we looked at ClinGenGene, and it pulled back 43 variants that occur within genes related to cancer phenotypes. The most impactful of those variants was a frame shift deletion in PALB2, PALB2, and that PALB2 is pretty well known to be related to a variety of cancers: ovarian, colon cancer, breast carcinoma. So that might actually be interesting to know that you might have some kind of predisposition, but the variant isn't overtly known to cause predisposition. We're just kind of assuming that based on what the gene is related to. There were additionally 25 other genes that contain missense variants related to various other cancers. But any given germline missense variants may or may not be conferring any significant risk for cancers. Hard to say. So what then did we find from PharmGKB? Again, this is a database looking for variants related to drugs, basically. It first identified an intronic variant associated with drugs for major depressive disorder. And it second found a UTR variant associated with methotrexate, which is both a chemotherapy agent and an immune system suppressant. For some reason, I expected to find many more PharmGKB variants than these 2. So not only that there was only 2, but only one of them related to cancer was, for me personally, a little bit surprising. But if we take these informations and like we put these pieces of data all together, what do they collectively mean for a single individual who's basically phenotypically normal? Well, it's seeming apparent that the clinical utility of findings from some random genome are pretty limited, especially to be reported back to them. It's possible that if they do get cancer at some point and they receive treatment for their cancer, it might be relevant to know that they have a variant that makes it like have lower efficacy or have an increased risk of adverse outcomes. That has some utility. But I would say in general that having a person who we know is at an increased risk to, let's say, familial cancer because we see it in their family history might be a better idea than picking a random person and trying to decide what's important enough to tell them. So for ClinVar, they had one variant related to hereditary prostate cancer. It could be used to inform, to encourage early intervention, but for many patients, that might not be worthwhile doing in the first place. More questions than answers. Same thing with ClinGen gene, although it's at a much shakier ground. This is a variant within a gene that is associated with it. But how frequently are these Pol-B loss of function mutations occurring in the natural population? Do they really confer risk is a question that would be need to be teased out before telling the patients anything at all. Case study 4 is, let's go on to case study 4. It's occurrence of somatic mutations within molecular subgroups among 182 patients with AML. So similar to case study 2, we have variants and mutations from many, many, many patients, except the idea here is that we can group them to find meaningful patterns. Particularly for heterogeneous cancer types like AML, clinical subgrouping can be very helpful for informing prognostic value, the prognostic value of their mutations. So there's this paper from Papa Emmanuel et al. That assessed driver mutations among 1,540 patients and sought to identify genomic subgroups. And then further, if there are prognostic implications for those genomic subgroups of their somatic mutations. They found more than 5,000 driver mutations among 76 different genes and regions with more than 2 drivers in 86% of their patients. So it's very widespread, but there are some patterns that can be teased out, meaning that molecular subgroups aren't 2 or 3 groups. They're, I don't know, a dozen or so different groups, but they all have distinct driver lesion patterns, and then they have distinct prognostic implications as a consequence. And that this can provide a better understanding of how the cancer is formed and the patterns of co-mutation in particular are relevant for their prognosis rather than any one particular variant. So this plot here on the right shows along the top diagonal how many people had one of the categories, and the internal nodes in some shades of blue indicate the number of patients that had co-mutation of multiple risk factors. And those people with co-mutations really needed to be set apart separately. So we wanted to see how these rules actually shake out in a separate cohort of data. So we obtained data from the Cancer Genome Atlas on the Genomic Data Commons for 182 patients with AML. Unfortunately, there is a whole class of variation that are not covered by OpenCravat: large inversions, translocations, gene fusions, massive copy number variants. They have to be called separately. And as soon as we get the funding, OpenCravat will be expanded to cover these kinds of variants. So let's get into the actual details. What is a genomic subgroup 1? It's people with NPM1 mutations. NPM1 is one of the most frequently mutated genes in AML, and it confers a relatively favorable risk compared to some of the other subgroups we're going to get into. So of all of the samples that we had, 182, 22 of them have NPM1 mutations, and 2 of them were seen more than once. So they're very widely distributed what kind of NPM1 mutation you might have, but they are relatively common in our data set. Subgroup 2 is AML with mutated chromatin, RNA splicing genes, or both. They defined a list in the paper of relevant genes from RUNX1 to 0SR2. And through the filter interface, it was pretty straightforward to input all of the samples all at once into OpenCravat, do a filter based upon protein coding variants that impact this list of genes. And we pulled back a total of 17 protein coding mutations that affect these genes, and 3 of them are seen more than one time. So these might be prognostic indicators that can be found recurrently in multiple samples. But again, we see that it's pretty widely distributed. There's not a lot of commonalities amongst these 182 folks in terms of their particular mutations. Genomic subgroup 3 is AML with TP53 mutations. And we know that TP53 mutations tend to have much poorer survival. And there are 37 patients out of our 182 that do have TP53 mutations. As is frequently seen in TP53, some of these variants are recurrent. So those are listed down below. The paper on the right indicated with the link down below shows the importance actually of this genomic subgroup, because the combination of TP53 mutation and a complex karyotype occurring have really severe consequences on the prognosis of a particular patient. Genomic subgroup 4 is CEBPA mutations. We were unable to observe any folks in the 182 who had biallelic CEBPA mutations. And genomic subgroup 5 is IDHR2 mutations at the 172th position. They say in the Papa Emanuel paper that although these variants are rare, they have pretty severe, striking, let's say, consequences compared to 140 mutations. So we did identify 4 patients in the subgroup with variants at R172K, but not at the other alleles of R172. So this is definitely something that occurs recurrently among patients. So there's a lot of details here, but if we zoom out a bit, we can find some commonalities, some findings. Out of 182 total patients, using the genomic subgroups that we can use using OpenCravat, 76 of the patients were able to be grouped. That means that more than 100 patients have some other mutations or some other larger scale mutations responsible for the progression of their disease. We observed that 11 of these 49 total mutations occurred in more than one patient. So although at first it seemed like they were very widely distributed, there is some repeated occurrence of variants, at least in this 182 patients with AML. So there is significant value in identifying patient subgroups, as was illustrated by those survival curves. They're tremendously different from one another in the outcomes, at least statistically. And there's also the potential for some patients to harbor more than one relevant alteration, especially as they progress in the disease, things tend to accumulate. So there is some potential that the signatures may not encompass the pairings of mutations, the co-mutations that I mentioned earlier. And particularly for underrepresented groups, they may have a slightly different presentation. So I hope that taken all together, these case studies not only motivate the use of OpenCravat, but the use of variant annotation tools and the importance of taking into consideration many different lines of evidence to prioritize variants that are relevant and important for further study. There are many opportunities for collaboration, so please do reach out to us at support@cravat.us or to me after this presentation, and I'd love to give more detailed demos of the software. Thank you.
All right, thank you so much, Dr. Pagel. I'll turn it over to the audience and see if anybody has any questions for her. While they're thinking, I was thinking myself about how we're very phenotypic driven, and I wondered if there's apps in the App Store that can be integrated that would kind of focus on or integrate the phenotype of the patients. Like if you use your original ovarian cancer example, could you say like high-grade serous, and then it kind of picks more variants that might be linked to that, or ovarian cancer in general?
That's something that has been a little bit tricky to implement, but we have recently included the human phenotype ontology. So rather than adding phenotype as an input, it can be used as a filter instead to more prioritize those variants.
Okay, so more like if it were to appear in the ClinVar annotation, then you could kind of filter by the phenotypes seen in ClinVar, for example.
Yeah, exactly. Yeah. You can search among the variants that are returned in ClinVar, in ClinGen, or based on genes with HPO terms that are relevant, so that it still has to be semi-manual. That's all I can say. We don't have a great phenotype ontology in things like ClinVar yet.
Yeah, great. Yeah, I know that's kind of the next step in a lot of these tools is integrating that next level of information. Anyone else have a question for Dr. Pagel?
One question, Kim. So really great talk. I just wondering, like in your case too, for the NDX network graph. So could you tell a little bit more about that graph? You know, is that predicted a relationship, or is actually validated by the publication already? I'm not sure.
So,
yeah.
That is a series of cancer pathways. I think they're pretty set in stone from the NCI. You'd have to go back to the particular set of networks put out by the NCI to verify the relationship there from that perspective. I'd have to pull back up the publication. But yeah, I can't answer on the exact. I wish I could, actually. I'm going to read into that.
All right, thank you.
Anybody else? Still got a few more minutes if anybody has a question. In terms of the input, it looked like you could put in VCFs, but then also add the BAM files. Is that correct?
We can visualize BAMs with IGV. The active variant calling is an incredibly fiddly process that we are happy to put onto the users, and they have centers at most universities that handle this. That way it's being handled properly, as there are lots of very specific fine tuning that must occur based upon what kind of BAM you've got, if that makes sense.
Yep, and then in terms of finding those somatic mutations, they're primarily done from the tumor, like sequencing of the tumor samples, correct? Versus like low level from blood or other samples.
Most of the variants that we've used from somatic are honestly the foundation reports. That's the data that I'm seeing most frequently. And sometimes it's sequencing of the samples, but that data is a little harder to get.
Okay, all right. Well, I think we'll thank you then so much again for the great presentation, and I look forward to kind of digging into the tools. There's a lot there to look at, so appreciate yeah, you spending your time sharing those with us today.
Thank you,
Thank you.
everybody.
Thank you.
Bye.